Hello, my name is Cesar Castillo. I'm going to be your instructor for Biology 11 this summer. I'm very pleased to have met many of you through email correspondences already. Thank you for sending the emails with your concerns, problems, questions, comments. Uh, Please feel free to contact me. I have another job in the morning, so I will try to get back to you as soon as I can, but hold on tight. I will usually hold off and respond until after 3 o'clock in the afternoon. So the plan for the semester, as you'll see today, is that I'll be posting uh, videos here on Blackboard Collaborate sometime after 3 o'clock. So I'll try to make it around 4 o'clock in the afternoon or 3:30, and no later, and not start anytime later than 4:30. All right. But the beauty of it is that you can access these videos at any time, any day. All right. So they'll be recorded, they'll be left on Blackboard throughout the rest of the semester, and you'll be able to access them whenever. Okay, now, let's see. So in this slide, you'll be able to see the updated syllabus. I just wanted to go over that with the class through here, and basically we'll start off with what it's telling you here. Of course, you should all be aware of what the lab manual is. It's the Essentials of Biology by Sylvia Mader. I think it's the 5th edition, and the edition itself doesn't quite matter. Many of you will have some older editions. I may not even have the same edition that you have, and the end result is going to be the same because most of the questions will be the same. The main differences between the editions are the exercises, which we are not really going to be able to do thanks to the coronavirus. So we're going to basically really concentrate on the theory behind these chapters in the book. There'll be some questions that I will choose that you guys should get to know. And as far as filling out the lab book, do the best you can. So due to the coronavirus, I'm only going to grade the lab books based on completeness. So if you show that you've done it, then great, and you'll get your points at the end of the semester. So I'll draw your attention to this here. Okay, there are supposedly on the syllabus 2 times that I'll be grading your lab manuals. All right, and if you just complete your lab manual as best as you can, it'll be you'll be given 5 points right away. All right, so that's a total of 10 points. That's the difference between an A minus and an A plus. Excuse me, B plus and an A, right? So it's a big difference. Make sure you guys just do the lab book as best as you can. I'm not looking to penalize anybody for getting something wrong or forgetting to fill in something here. And as long as you showed that you filled out as much as you could, then you're going to get the points. And as I go through these PowerPoints, I'm going to show you exactly the parts that you need to fill out. And hopefully you can match that up real well with your lab manual. All right. Now, there's also the lab quizzes. Lots of questions based on the lab quizzes. All right. So the lab quizzes are from the website that you're linked to through the Darwin to Plato, Plato to Darwin to DNA book. So like I had said in the emails, make sure that your book is new, that the code on the inside front cover of the book is new and unused. If you're trying to buy a used book, then you can't use that code, or it'll link you to the last semester's quizzes or for something like that. All right, so you need a new code for that, and you need a new lab manual also, because you should not have somebody else's answers. All right. If you have a physical copy of these books, not a big deal, right? In fact, it's great. And when it comes time to handing in the lab manual, you can take pictures or scan it, and then I'll show you, if you don't know already, how to make one large PDF uh, uh, file, one large PDF file, so that it's easier for me to receive it and for me to grade it and for you to get it back or send it. All right, and there are some free online services that do this. Okay, so there are 4 lab quizzes. You should have, you should be granted 2 attempts. I know there's an issue with that now with some people and possibly everyone. I'm not sure. I'm looking at the settings that I have placed through my end, and it looks like it has 2 attempts. So hopefully it looks good for everyone else as well. So you should have been granted 2 attempts. That is, you can take the quiz once, and if you don't like the grade, you can take it again the second time. Both times, you're given a certain amount of time. I think it's 30 minutes, right, which is more than enough time. And the lab quizzes are based only on the Plato to Darwin to DNA book. So they're not based on the lab manuals or the lab exercises. The other thing, the lab exams, that's this one here, this part. Okay. There are 2 lab exams, one in the middle of the semester and one at the end of the semester. And the lab exams are usually open, not multiple choice, but more of a sorry, not multiple choice, but just quick fill in. But this semester we're going to do a little bit of a change, and we're going to have you write a little bit more, like a sentence or close to a paragraph, and there will be fewer questions. But be aware that the exams are based on the recitations that I'm presenting and on the actual information that you find in the lab book. All right, so the 2 exams are probably going to be the most challenging portion of the lab, and make sure that you study well for them. All right, on how the exams will be presented to you, I don't have all the information yet, but I'll get back to you as soon as I clear that up with Dr. Muehlbauer on how she wants it to be done. All right, Dr. Muehlbauer being the course head for this class. Okay, I don't believe there's anyone registered under me under Bio 10, but just in case. Okay, so Bio 10 is another class that you just take only as the lab portion, and you don't have to take the lecture. But that only applies for people who have already taken the lecture at Queens College many years ago, right? So you should not be registered as a Bio 10 student unless you've already done the lecture under Bio 9 sometime in the past. So if you're brand new to this course, you should all be taking Bio 11. All right, so take a look at the left side or the first page of the syllabus. And I think I explained some of this in the emails. All right, so today, Monday, June 1st, we have Lab 1, which corresponds to Chapter 1. All right, in this book, right, in the Mater text. All right, and the Plato to Darwin to DNA book is the Muehlbauer, Chapter 1. We don't really discuss that book in lab because it's more of a lecture thing, although see, in the past, we would have you write an essay on the book at the end of the semester. But this semester we eliminated that. Okay, so only the quizzes are the only thing that links the lab to this book written by Dr. Muehlbauer. So make sure, all right, that you do have the lab manual available to you at some point before the end of the semester, hopefully halfway through the semester. I know that it's very difficult for some people to get it shipped right away. But the beauty of this is that, like I said before, these recitations where I explain what it is that you have to know about the book and what you have to fill out, uh, uh, you will have to, you'll be able to fill it in later on. And you can see these recitations as you go. Okay. Now, then tomorrow, I'll be posting a recitation on lab 2, which is the in the lab book. It's metric measurement and microscopy. So that's actually chapters 2 and 3. So just be aware, that's chapters 2 and 3 for tomorrow. And then the day after, we're going to be doing chapter 4, even though it's lab 3. All right, and so on. So lab 3 has chapter 4. Okay. Now, let's move on to the next slide. Here is another file that I sent to you by email. It's just a simple calendar to keep track of where we are. But again, it's kind of the same thing as the syllabus. Today is June 1st. We're doing chapter 1. 1 is the scientific method. All right, and then June 2nd, we're doing number 2, which is this. Okay, and it tells you the title: metric measurement and microscopy. So just something to keep track of. All right, nothing too dire if you don't follow it. All right, now moving on. Okay, so I actually wish that the lab manual was available as an ebook. That would be wonderful. Okay, but I can't seem to find it. But if any of you do find it, that would be great. Okay. But if you don't receive it anytime soon, don't worry about it. Okay, the lab manual, you don't have to hand it in until later on. All right. Okay, so in today's lab manual, in today's chapter, the topic that's covered is the scientific method, and the book uses a biological entity known as the pillbug or Armadillidium vulgare, or the common pillbug, as an example or as a tool for you guys to learn about the scientific method. And I always start this class out by explaining a lot of these things that you guys have been exposed to throughout your careers as students from elementary school on up. Okay, and basically what we have is a system that allows for the entire scientific community to be able to agree on whether or not your findings for some question that you may have had are valuable, and if they're valuable, can the other scientific researchers repeat them, copy them, and get the same results. So the scientific method is just a way to get everyone to be able to approach or get up to the same conclusions if they follow the same steps that you took. So you can start off with an observation. An observation, you see something and you're curious about it. You're wondering, hey, I wonder why this happens or how it happens, etc. So you formulate a question, and the question is something that you'll say, well, the sky is blue, right? That's your observation. Why is the sky blue? Well, that's your question. Why is the sky blue? And maybe you formulate a hypothesis. An hypothesis. Your hypothesis should be, okay, so your hypothesis should be well informed, right? So you're not going to say something crazy like, oh, the sky is blue because it reflects the color of the ocean. Well, that's easily testable because you can go to the largest landmass, which is Asia, and find the center point and look up into the sky, and it's still blue, even though you're nowhere near a large body of water. But that was a not well-informed hypothesis. What you want to do is you want to study up on the question. You want to do some research. You want to find out, hey, what have other people thought about this question? And it could be something, complex. Your hypothesis. What your hypothesis is going to do is it's going to attempt to answer your question in a way that you'll be able to create a prediction. So like what I said before, right? Your uninformed hypothesis where the sky is blue because it reflects the color of the water, it was easily testable, right? You were able to predict, well, if that's the truth, then the sky should not be blue when I'm very far away from water. So that's a prediction that you're able to make, and that prediction will be used to test the strength of your hypothesis. Okay, so your hypothesis can be, you'll have your what we call the alternate hypothesis and your null hypothesis. So in my example of the uninformed hypothesis, your alternate hypothesis is the sky is blue because it reflects the color of the water. And your null hypothesis is the color of the water has nothing to do with it. And in this case, we found that the null hypothesis stands up and we reject the alternate, or the alternative hypothesis. Okay, so your hypothesis, like I said, should be informed. Okay, and you should be able to make predictions from it. And the purpose of these predictions is to be able to support or reject your hypothesis. All right, do you accept your alternative hypothesis, which is what you think, or do you accept the null, which is the opposite, right? Such and such has nothing to do with that, for example. Okay. All right, so your first hypothesis, you threw it out the window. You have a second hypothesis. Well, maybe it has something to do with some particles up in the air reflecting light from the sun, right? As it turns out, that's kind of how it works. Basically, you have enough particles in the sky that reflect everything except blue light. Well, blue light is reflected. I apologize. The blue light is reflected off the particles, and that's why we see the sky is blue. All right. So You have your hypothesis, which helps you make predictions. The predictions tell you whether, once you have your prediction, you test your hypothesis. And the testing will, if it matches up with your prediction, then your hypothesis is supported. If it doesn't stop with your prediction, it comes up with a surprising answer or something unexpected, then your hypothesis usually gets rejected, right? Depends on the situation. And your null hypothesis stands. The one that says, no, my original hypothesis does not explain the answer. All right, so that's what the importance of testing is. So this is basically your experiment, right? Experimentation. So that's the important part. Everybody knows about science has experiments. Well, that's what testing is. Your experiment. You create an experiment that will help you figure out whether your hypothesis is valid or not. All right, so you end up with some sort of conclusion experiment, and if the conclusion is something that you think is worth communicating, then you do so, especially if you support your hypothesis. If your original hypothesis stands up it looks to be correct, then you end up writing a paper about it. You communicate it to the rest of the scientific community. All right, so all right, so one thing that you should note, all right, let's see, I have it in the next slide. Right. Like I was saying before, your hypothesis must be testable because it has to be something to falsify. It has to be something that you can potentially prove to be false. All right, you can prove it to be false. You can prove it to be false, but not prove it to be true. All right, so this is especially in biology. If we were talking about math, you can prove something true in math, or you have some physics, for example, that you can prove, and chemistry, you can prove some stuff true. But in terms of biology, there really isn't anything out there that you can prove as absolutely 100% true. What ends up happening, though, in a lot of cases in science is you may have a hypothesis that goes under many, many types of experimentation over many, many years through many, many, many different people or research, and no one has been able to prove it false, at which point your hypothesis can become a theory. All right, so a theory is a hypothesis that has not been proven to be false. And again, like I said before, hasn't been proven to be false even after many years of experimentation through many, many different people. Many different researchers have attempted to find an alternate hypothesis to your hypothesis or to the hypothesis in question, and they failed. For example, the cell theory of life, right? The hypothesis is all life forms must be composed of cells, right? The unit of life being a cell. That hasn't been proven to be false yet. All right. Now, that's not to say that so it's a theory today, but it's not to say that it's not going to be proven false ever. Maybe someday someone will say, well, yeah, viruses, even though they're not made of cells, right? The coronavirus we have right now is not made of cells, but it is alive. Well, we don't know that. Right now, as it is, everybody believes viruses are not forms of life, uh, and therefore the cell theory holds for now. And it's not to say that somebody might not be able to convince everyone else opposite of that in the future, but it's unlikely. Uh, Another theory that comes to mind is the theory of evolution. Right, Darwin and a fellow by the name of Wallace independently came up with the same ideas of how evolution works, the theory of evolution through natural selection. And so far, no one's been able to say, hey, natural selection is not the way that evolution works. No one's been able to say that yet. It's not that it won't be true, that somebody won't be able to say that in the future. But for now, no one's been able to prove that natural selection is not the mechanism by which evolution occurs in living systems. All right, even though it's been over 150 years, I believe, since the theories came out. All right, so let's move on to the next slide. Some details on the experiments. The experiments themselves are very important. So your experiment is very, very important in terms of how well your hypothesis is accepted by the researchers in the scientific community or by other people in general. You don't even have to be a researcher or a scientist. You can be just any old person that's listening to the radio and listening to you speak about your experiment and your findings on the radio, for example. All right, so what are you looking for? Say, for example, somebody says they've discovered the cure for a common cold. Well, you're looking for a few key things. All right, an experiment is much more valuable, or also much more likely to actually be true. Their findings are more likely to be true if they have a very large sample size. So, in all experiments, you have a sample of people, or a sample of other living organisms, or whatever it is, a chemical or anything. And that sample, the sample has to be random. So, at least, for example, in biology, if you're testing a population of organisms, say you want to do an experiment on crab spiders that live in the Atlantic seacoast, you can't possibly go out into the ocean and find every single Atlantic crab spider and perform your experiment on them. So what you do is you take a sample, a population sample, and your hope is that your method of collecting these samples was random enough that that the cumulative effect of all the samples you've collected will represent the true population. All right, so in order for that to be true, you have to have a large amount of samples. The larger your sample size, the more likely it is that it represents the actual population. And you don't want to just take it from one location. You want to take it from several locations. So you're randomizing your samples. And if you do these two things, large sample sizes and random samples, the value of your experiment is much higher. It's more likely that the findings that you get are truly representative of what is going on in terms of what your hypothesis might be. Also, you want to have something that we call a negative control. Okay, so a negative control is basically whatever experiment you're conducting, you have a certain group from your sample that you are not putting through the steps of the experiment, at least not all the steps. All right, so for example, I'll give you the definition: a sample or event that is not exposed to the testing procedure. So you may have the sample go through almost all of the steps of the procedure, except the one key important part of the test, the part of the test that you're really concerned with. Right, so that's a negative control. All right, so for example, you'll have, say you're testing the effectiveness, the efficacy of a new drug on people. You're going to your sample, right? You're going to hopefully grab as many people as you can to participate in your experiment, and you're going to be as random as you possibly can. And some of those people are going to get something that looks like the medicine, but it's nothing more than a sugar pill. So that's a negative control. All right. So you're giving them something you know the outcome is, and the reason for this is that you want to compare the negative control to your actual experiment, to whatever it is you're testing. And if the results for the two groups are the same, then you know your thing is not really doing what it should be doing. So for example, if your drug has no difference in effects from the placebo, the sugar pill, then your drug is no better than a sugar pill. So it's no good. Okay? And like I've been saying before, the experiment must be repeatable. So you take meticulous notes on what you've done, and that way, other researchers will be able to replicate your results. They'll be able to take the steps that you took. Sometimes it can be very sensitive. What was the temperature in the room? What was the air pressure? What was the temperature of the samples? What were the steps? Did you take this sample first and then that, put it through water, then through alcohol, or whatever it is that you want to do? So everything has to be specific, very specific, so that other people can say, oh yeah, that guy's not lying. He's not pulling this out of thin air, this information out of thin air. I did the exact same thing that that person did, and I got the exact same results. Yeah, that's true. All right, so an experiment that is repeatable is extremely valuable. Okay, so I cut off the E. All right, so if you have a question of what a random sample looks like, so here in red, circled in red, I have my original population. So for example, this could be the entire population of pill bugs that can be found on the campus of Queens College. And you want to do some sort of experiment on them. Well, you go out, and do you really want to capture them all from the same spot? No. So what you want to do is you want to go to different parts of campus and collect from these different locations. So you're not going to collect all the pill bugs you see. You're just going to collect some of them. So you take, for example, you take this one from behind Powdermaker Hall. You take this one from behind Colwyn Hall, and that one from behind Clapper Hall, and this one from behind the library. All right, something like that. Very simple explanation of what random samples should look like. All right, and like I said before, random samples help increase the power of your experiment. I'll give you an example now of, I think I've been giving you this example all along. So for example, say you are a scientist in a pharmacy or a pharmaceutical company, and you created a new drug to treat, hopefully, the common cold. Be nice if somebody here in this class eventually created something like this. All right, so you are going to test your drug because you want to see if it works, right? You want to test your drug on as many people as possible. So if you're that citizen in the car and you're listening to the scientist brag about his experiment, and he said he tested it on 100 people. throughout the country. Let me take a step back. He tested his new drug on 100 people only living in Nevada, right? And they all agreed that yeah, they um, they got cured by this. Are you going to believe that? Well, hopefully not. There could be some other thing. Why are all these people only 100 people in Nevada? I don't know. Well, maybe they smoked something before they took the pill, and so they felt better. But if you do, if you hear something like that on the radio, don't believe it, right? Or on TV or whatever, Twitter. Uh, However, if the experimenter that's announcing his results, his or her results, says, "Well, I tested this on 5,000 participants, which were a random sample from all states, and you have a good sample size, right?" And performed a double-blind study, which is basically the placebo. A double-blind study is the person giving the drug doesn't know whether the drug is a sugar pill or the actual drug, but at the same time, the person receiving the drug doesn't know either. All right, so that increases the ability for the experiment to be reliable. And the whole purpose of this is to reduce what we call bias. Okay. Sorry, that's my wife's phone. She forgot it here in the bedroom, my new office. Anyway, okay. So the double-blind studies is an example of a negative control. It's a particularly good negative control. All right. So if you find, here's the key word. If you find a significant amount of participants are cured, then you know that your drug is effective. All right, so how do we figure out what significant is? Significant is a word that scientists don't just bandy about randomly. Significant is a word that's used very specifically. So whenever you hear on the news or on Twitter or whatever outlet you use, somebody say significant, and if it's a scientist, there's some actual strength to that word. All right, there's a reason why they're using it. Usually, what happens is they use statistics. Okay, statistics basically just different mathematical methods, which we don't go into in this lab, in this class. My other class that I teach, we do a lot of statistics. But in this class, it's good enough for you to know that statistics help you remove bias. And bias is just what you expect the results to be. You know, so if you have bias for something, you expect something to occur, and you may interpret the data in a way that support your bias, support what you expect. So you want to remove that possibility, and you do that by statistics. Statistics will basically just use numbers so that you're not guessing as to whether or not your results are reliable or not. So there's a specific meaning to significant, right? So usually, and it has to do with the math of statistics. Okay. So for example, in my other class, we do one of the first statistics experiments that we do is we measure the heights of the males in the class versus the females in the class, and we do this statistic called the t-test. And at the end of the test, we're comparing the value of the number that we get to a table that was created decades ago. And if that number is greater than or equal to or greater than that value on the table, then we do have a significant difference, and we're able to say yes, the heights of males is usually larger or taller than the heights of females. So there was a significant difference in the heights of men and women. Okay. All right. So move on. All right. Ooh, I wonder why this turned out to be the. Okay. So armadillidium, the M got moved. Sorry about that. I don't know why that happened. In my original PowerPoint, it looked fine. All right. So armadillidium vulgare is the little pill bug that they talk about in class. In the book. And that's the little organism that we were going to use today in person had coronavirus not occurred. Yeah, I think that's the problem, that I didn't create the PDF. I usually do, and I just said it was such a small PowerPoint, I figured it wasn't worth it. But thanks, I'll keep that in mind. All right, so if we were in person in class, we would be playing around with these little guys. Okay, so there is a whole paragraph dedicated to their biology in the chapter, although I don't think it's the main point of the lab. The main point of the lab is the scientific method, but you should know a little something about them. All right, so Armadillidium vulgare, or the pill bug, or the roly-poly, find them in any garden. All right, you can find them in potted plants at Home Depot, for example, or you can find some other species related to them. And they're detrivores, which means that they will feed off dead material, usually plants. Okay, so if you try to keep them in the lab or at home as pets, you just put some leaves, some veggies, a couple of leaves of veggies in there, keep them nice and humid, and you'll have a whole colony of them. They are terrestrial crustaceans, which means they're living on land, and they're closely related to lobsters, shrimp, and crabs. You know, those things that people eat from the ocean. They have gills, like the crabs and the shrimp and the lobsters. So even though they're terrestrial, they have to live in very moist environments, like the soil, underneath the leaf litter, underneath fallen leaves and things like that. If you have a very clean garden, which tends to stay dryish, you may not have pill bugs. But if you're lucky enough to have a garden, if you have a garden it's a little bit messy, you probably have lots of them. All right, and of course, all crustaceans, crayfish, lobsters, shrimp, roly-polies, are part of a group known as the arthropods. And the arthropods include spiders, that's one group. The crustaceans is another group, and you have the insects as another group, entirely different. But they're all members of the Arthropoda phylum. And I'll discuss more about this towards the end of towards the beginning of the second half of this semester. Okay. So one of the things the book points out is they have a hard exoskeleton. And because it's a hard exoskeleton, in order for them to grow, they must shed the exoskeleton. So you've probably heard of molting. Spiders, some people keep tarantulas as pets, for example. Every now and then the tarantula will look like it's dying, and it turns around, and then all of a sudden the owner thinks it's dead. Well, what's happening is it's trying to crack out of its shell and come out of it and grow a little larger. All right. And so this is how all arthropods grow. They leave their exoskeleton and they pump up their body fluids in order to grow to a larger size. All right, so that's what all arthropods have in common. They have an exoskeleton that they shed in order to grow. And I thought I might have a little bit of fun so that you can remember this little guy. This little guy, which is relatively harmless, has cousins that are, well, let me discuss the cousins later. So here in this upper picture, you can see relative size of the pill bugs. They're completely harmless. If you do have a garden, why not? I think a lot of us did play with them as kids. They're called roly-polies because if you scare them a bit, they roll up into a tight little ball in which you can roll around on a surface. Their anatomy is not that important for this class. It really isn't. But I'm just presenting it here so that in case you wanted to be meticulous about your lab book. Okay, so they do have 7 pairs of jointed legs. The number of pairs of legs is important when it comes to arthropods. So, for example, centipedes have 1 pair of leg per segment. Here's a centipede, right? Each one of these is a segment, and each one of these would have a leg, 1 pair. Okay. But so that's what I mean by segment. So here's a segment, here's a segment, here's another one, here's another one. Okay. That's also a segment. Each one of these red lines represents a segment. It's a repetition of a body part. Right? Remember that because we're going to be discussing what segmentation is later on. And when it comes to arthropods, the number of legs per segment is very important. So for insects, for example, you have 3 pairs per segment, with the segment being the thorax. And in spiders, you have the head and the thorax fused together. That's the cephalothorax, and you have 4 pairs of legs. Millipedes have 2 pairs per segment. Centipedes have 1 pair. So whenever you are taking a science class and they talk about scientists, you're talking about organisms, the scientist writing the book nerds out on you and starts talking about the nitty-gritty of it all. All right. So 7 pairs of jointed legs. Sorry, and they also have these little other appendages, which are called the pods, which are used for reproduction. And underneath this, there's a little casing if it's a female, which holds on to some eggs. Not dissimilar, very, very similar indeed to, say, a shrimp holding eggs. All right. If you don't know what I'm talking about, quickly Google shrimp with eggs. You should be able to see videos of that. Or pillbug with eggs, right? So I thought I might have some fun with the pillbug and show you some examples of their cousins. So the sea louse is like a giant pillbug, and apparently they're delicacies. It's somewhere on this planet. They'll trawl these out of the bottoms of the oceans, and they'll sell them for food. You boil them up like or something. And there are others that are more menacing. So there's a species that is parasitic, and what it does is that it goes in through the gills, right? Swims up through the gills right here of a fish, climbs into the back of the mouth of the fish, and it will, basically cut off the blood supply to the tongue or to whatever is there inside the mouth of the fish, and replace it. Whenever the fish takes in food, the pillbug gets its fair share. Well, not the pillbug, but this louse gets its fair share. So they're varied. There's a lot of variation in biology. So that's the point I'm trying to get across. You have a common design, and by the diversity amongst living things. You take that one design and you change it up a little bit in every now and then. Okay. All right. So at this point, I'm going to share my screen because I want to go through the lab manual. Okay. All right. For those of you attending, hopefully you can see the lab manual. Let me know. Just type in yes or no, or I'll just keep going. Great. Okay, thank you so much. All right. So here on this page, I have the lab manual. All right. So this, like I said before, may not be the exact same version that you have, but it's good enough. All right. So there may be a question of something like this: Why does the scientific method begin with observations? All right. So what's up with the observations? You can't have a hypothesis on something you don't have any curiosity over, right? Observation is the first step of the scientific method. All right, first step. All right, formulating a hypothesis based on their observations, scientists come to a tentative decision. All right, called hypothesis. This is a tentative, I wouldn't say decision, a tentative explanation. So a tentative explanation. Forming hypotheses help scientists decide how an experiment will be conducted. That's true. Okay. So in the lab, you would have exposed the pill bugs to some food items, for example. You would have given them some applesauce or soda, Coca-Cola or Pepsi. And your hypothesis would have been, for example, if I was doing it, I would say my hypothesis is the pill bug would be attracted to the applesauce because it's plant-based, as opposed to the Pepsi, which by the way is acidic and kind of, I know my children run away from it because they don't like the feeling of the spiciness inside their mouths. So something like that. All right. So you will formulate a hypothesis, which helps you make a prediction, and that prediction helps you formulate an experiment. So my hypothesis, I think pillbugs like applesauce better. And so that's my prediction. Experiment is okay. Fine, I'll make an experiment. Well, I'll expose a pillbug to both applesauce and Pepsi, and see what happens. All right. So that's the benefit of formulating a hypothesis. Think of something to say here. All right. Something like what I just said. Doesn't have to be too complex. All right. And I explained what a negative control was. Here's one part I forgot. Scientists call the results of their experiments data. All right, so make sure you remember the term data. Data is just the quantitative information that you gather after your experiment. Right. So the data could be continuous, or the data can be unrelated to each other. So you could have a continuous data would be values like 1 through 10, somewhere in between, or you can have the opposite of continuous, which would be this one is male, that one female. This one is I don't know. This one behaves in this format, and the other one behaves in another way. So you can have all those different types of data, but you don't need to know the different types of data. So don't worry about that. Okay, when another person repeats the same experiment and the data is the same, both experiments have merit. Why must the scientist keep a complete record of an experiment? Because the experiment must be repeatable, right? It has to be something that other people can do. Repeatable. Coming to a conclusion. Scientists come to a conclusion as to whether their data support or do not support the hypothesis. If a pill bug is attracted to fruit juice, your hypothesis is supported. If the pill bug is not attracted to fruit juice, your hypothesis is not supported. A scientist never says that a hypothesis has been proven true because, after all, some future knowledge might have a bearing on the experiment. What is the purpose of the conclusion? So the purpose of the conclusion is to support, right, or reject your hypothesis. Your hypothesis, the one you're supporting or rejecting is, remember, is the alternative hypothesis. Alternative. So remember, if you support your alternative hypothesis, you reject your null. If you reject your alternative hypothesis, then you accept your null hypothesis versus null. Okay, developing a scientific theory. A theory in science is an encompassing conclusion based on many individual conclusions in the same field. For example, the gene theory states that organisms inherit coded information that controls their anatomy, physiology, and behavior. It takes many years for scientists to develop a theory, and therefore, we will not be developing any theories today. Most certainly not. How is the scientific theory different from a conclusion? All right, so it's undergone, I would say, rigorous testing. And by rigorous testing, I mean many different researchers have tested your hypothesis, and it's been tested for many years under different types of experiments, in different ways of testing the same thing. All right, so in this flow chart, which is kind of the same thing I showed you before, a little bit more complex, you have your potential hypothesis, which is step one. You can have other hypotheses as well. All right, one of them can be your alternative. You can have two alternative hypotheses, and then your null, or whatever you want, right? And your hypotheses lead to a prediction. The prediction helps you create an experiment, because once you have a prediction, you can test it. So the experiment is for testing. And then at the end of the experiment, you can either accept or reject your hypothesis. If you reject it, you go back to one of the other ones. Your null hypothesis, or if you have more than just two hypotheses, you have something else to go into. And you repeat and repeat and repeat and so on until you're finally satisfied. Okay? And eventually you are led to a conclusion, and with that conclusion you can move on to sharing your information conclusion with other people. Okay. So in your lab books, observing a pill bug, do not worry about this information. Okay. do not fill in these parts where you would have been required to do an experiment or observation in the lab, because for obvious reasons, we're not in the lab. All right, so this is going to be, like I said before, mostly a course about the theory of the material in the book. All right. Don't worry about drawing a pill bug. The point that I have here will, you can get a drawing from online, so there's no point. All right. So the exercise here, why would they ask you to draw? Is it to kill time? No. The point of it was that this was an exercise in the powers of observation. Observation is a very important step in the scientific method, after all. So if not everybody has the same ability to observe something very well. They may miss some little detail here or there. So when you're doing science, observation is very important. You have to really have an eye for details. And so this was just an exercise in honing your skills a little bit. All right, and what's happening over here in this page, Pillbug's motion, which again, you don't have to fill out at all. Any experiments that have to do with the pill bug in your lab book, you can leave blank. All right, so we were going to put the pill bug through a series of gauntlets. So we're going to say, okay, we're going to put it on a flat surface, see how fast it can move. We're going to put it in a test tube, put the test tube at an angle or in different directions, and see which direction the pill bug prefers to move, etc. So it's just more exercises and observation to see how best to work with this animal. Okay, and like I said before, one of the things that we were going to do is see how fast it went, and we were going to have a ruler, and the pill bug. And the pill bug was going to move in some direction, hopefully the correct direction. And we were going to measure time versus distance, right? So we're going to be able to say pill bug 1 ran 20 millimeters in 60 seconds. Something like that. But don't worry about that. It's just for you to know what that was. We were going to get 3 samples, random samples, and average it out. And that's as far as we went with statistics in this lab. Just finding the average. Okay, so some of you may be thinking, why millimeters and not inches? Because in science, we use metric measurement, which is the units of measure that the entire world uses when doing science. And most of the countries on the planet use the metric system for everything, not just science, except the United States and a few other countries that use inches or the old English system or imperial system. But when you're doing science, and in this class, anything you write should be in the metric system. And tomorrow I'll discuss what metric system is. So, for example, the millimeter rather than the inch, or we're not using a quarter inch or anything like that. We're using millimeters and meters, etc. And in terms of time, we use seconds. Seconds are metric measures. All right. Now, formulating hypotheses. This is another thing we don't really have to do. Okay. So, like I said, we were going to expose the pill bugs to different food items. And the different food items were going to be, or not all of them were going to be food. Some of them were going to be something like baking soda. Baking soda versus, for example, flour. And baking flour. And your hypothesis was going to be which one of the two was it going to like. All right. And we were going to expose the pill bug to some control items, which were going to be for liquids, it was going to be water, and for solids, it would be sand. Because everything that's alive loves water. It needs it. Maybe it doesn't love it, but it needs the water. So, especially with pill bugs, we know that they exist in very moist environments because they breathe through their gills. Gills must be present, can only function in the presence of humidity, right? Of high environment of high water. Gills, when we speak about gills, we tend to think of gills only functioning underwater. But as you can see with the pill bugs, which are terrestrial, they can function in humid environments and not completely wet environments. Right, so the pill bug, what we know, is going to like the water. All right, and sand offers nothing for the pill bug. It doesn't hurt the pill bug. It doesn't provide anything good for the pill bug or bad. So we knew that the pill bug was going to either ignore the sand, was probably going to just ignore the sand. So we use as a control. And then the other items were going to be food items. So we're going to have soda and some sort of juice. Here we're going to have flour, which was plant-based, flour from wheat, and maybe baking soda, which is a kind of chemical, right? Which is a kind of a chemical that's foreign to living things. And you were going to formula a hypothesis. Dislike, for example, disliked, liked, neutral, and maybe liked, disliked, and neutral. And we were going to say reasons, this is plant-based. Plant-based, sorry, this one is plant-based. And this soda one is acidic, right? The flour is plant-based. And the baking soda is foreign chemical, right? So you can write something like this. All right. So that you can certainly fill this table out. You can copy what I wrote. I don't mind. And this you don't have to do, because this would have been the actual experiment. All right. And these would have been class results, which we certainly don't have. All right. Which brings me to this section. I'll stop sharing my screen. Okay. Guys, if you have questions, hold off for a few minutes while I finish this. Okay. All right. So the last portion of the lab chapter that we're going to cover is the laboratory review, which is some questions. And these are the questions that we'll use for the lab exams. So make sure that you do review these at some point before the lab exam. All right, so the first question is, what kind of an animal is a pill bug? You can say it's just a crustacean. Okay, let me see, right? Crustacean. Or arthropod is fine. Okay. What kind of skeleton does it have? We call that an exoskeleton. Okay, I have not yet developed good handwriting when writing on a computer screen, so excuse me. All right. Guys, hold off your questions until after I'm done here. What structures do pill bugs use for gas exchange? In other words, how do they breathe? Right? And I was just discussing those about a minute ago. Those were the gills, and they need humidity, right? What do scientists do first when they begin to study a specific topic? Well, the first step in the scientific method is observation. And what is a tentative decision? Again, I don't like that. I hope your books have a better word. Tentative explanation. And that would be the hypothesis. Right? If your theory is a thesis, theory being something that's really accepted, hypo is under. So hypo theory, underneath your theory. Hypothesis is the first explanation you have. So that's where we get that word from. What do we call the sample that lacks the factor being tested and goes through all the steps of the experiment? Well, we call that a control. Yeah, I know I can type it. I kind of like writing. All right. What do scientists call the information they collect while doing experiments or making observations? And that is data or data. However you want to pronounce it is fine. Which is made by scientists following experiments and observations, a theory or a conclusion. All right. So after your experiment, right? First, your experiment comes after your observations. After your experiments, what you come up with is a conclusion. Okay. Conclusion. And what do scientists develop after many years of experimentation? And a lot of similar individual conclusions. In that case, you have theory. Okay. And if your hypothesis was that your pill bug would be attracted to applesauce and your pill bug moved toward the applesauce, what would you say about your hypothesis? Well, in that case, you would say my hypothesis was supported, not proven, right? You don't say proven, you say supported. And here, for 11 and 12, indicate whether the statements are hypotheses, conclusions, or scientific theories. All right, all organisms are made of cells. Now, we know that is a theory. I discussed that earlier. It's a cell theory of life. And the data show that trans fat intake raises cholesterol and contributes to heart disease. That would be a conclusion. Now, if a pill bug travels 3 millimeters in 30 seconds, what is the rate of speed? It's very easy. 3 millimeters divided by 30, 10, right? 10 millimeters per second. So that's your answer. Simple math. If a pill bug moves towards a substance, is it attracted to a repellent? That's a dumb question. I hate that question. So we can just eliminate that. If you have something similar to that, you don't have to fill that. Okay. All right, next. Now here's some hopefully questions. Conclusion. All right, so this is something that's a good example of a question that would show up on your lab exams. And of course, the answer would be that a theory, right, has been tested more rigorously. More rigorously than a simple conclusion. So it's been through, you can say anything. You could say it's been through many, many tests, many, many experiments over a long period of time. over a long period of time. Right? Why is it important to have a control substance for an experiment? Okay, so control tells you whether or not your your substance is effective. Okay, so control indicates is a good word. Indicates whether or not the the whether or not the experimental subject. The experimental subject being, could be the substance that you're testing, for example, a chemical, a drug, the food item for the pill bugs, whatever, is effective or not. Oh, I said whether or not, so it's effective. We can just leave it there. Why is it important to test the pill bug's response using one substance at a time? Look, we're not responsible for something like this because we didn't do the actual experiment. Okay, but it's an easy answer anyway. You want to test one sample at a time so that you're not mixing up the conclusions for each sample. Okay, so I'm going to stop the recording here.